let's talk now about what you would really do, right? If you truly, really want to do calcium imaging, if your goal is, I want to know how these neurons are behaving, let's do some calcium imaging, right? I've got some GCamp viruses in the fridge upstairs. There's a 2-photon microscope upstairs. Let's get to it, right? How are we going to do this? Well, the first step is to get some glowy cells, right? It's to get some cells expressing GCamp. I mean, it doesn't have to be GCamp, but it's probably going to be GCamp. And how can we do that? Well, as I said, there's viral transduction. The great thing about viral transduction is you can get in an aliquot virus. It'll be enough to inject. Sorry, it'll be enough to inject 100 mice. Costs about $100. Bingo, bango, you're away. You've got to perform some kind of surgery. The surgery's not a big deal. You do a little tiny wee hole in the skull. You put in basically a very fine hypodermic needle. You inject in a few nanoliters of virus. You come back in 2 weeks, right? On the other hand, doing those surgeries is a bit of a pain. Some days they work and some days they don't work, and some days they work better in one mouse and not so good in another mouse. So having a mouse strain is nice, right? If your boss has lots of money, you should be arguing to get a mouse strain. So there's a Cree expressing mouse line. There's a GCamp with a LOX-P site on each side. You breed them together, you get the fluorescent where you want it. This is good for lots of reasons. It's much more standardized. You can skip that surgical step, but you've got to pay for it. Not just getting the mice in, but you've got to breed them. You've got to keep them going. You've got to organize this breeding. That can be a worse of a pain, depending on your institution, than just doing the viruses. On the other hand, sometimes there is no strain to achieve what you want, where you have to resort to viruses, right? So you could be interested in some weird cell in the hypothalamus and there's just no Cree line. So what you just have to do is just put your injection pipette into that nucleus of the hypothalamus and inject and cross your fingers. Then you need to get the animal under the 2-photon microscope. So you can do this as in sort of 2 ways. Easiest way is anesthetize. Maybe that you don't care, about how these neurons respond to behavior. You just want to know whether they're firing in response to a drug or whether they respond to, gross sensory stimuli. So you just anesthetize the animal in the morning. One of these ones that you've put the virus in or you've bred. You anesthetize it. You do the surgery to put in the cranial window. That might take an hour or 2. You glue on whatever head bar that's going to hold the animal still. You put it under the microscope. You get the imaging done by the end of the day. You can go home at 4 o'clock. Probably had a pretty good day. If, on the other hand, you want to look at how some neurons respond during behavior, then you better suit up, right? Because this is going to be a long haul, right? So you've got to implant a chronic cranial window, not just the one that's got to last for the day. You've got to put one in that has to be sterile and stable, and you've got to do it much more carefully. Then you have to train the animal to accept the head restraint, which can take up to a week. And then you've got to train the animal to perform whatever task you want to do while it's head restrained, and that can take weeks, right? And this whole time, something can go wrong. The mouse can get an infection. It's the head bar, the head fixation anchor can break off. The mouse can just not learn the task. Or maybe you can suck at teaching a mouse to do the task and you can't teach them to do the task. So it's going to be a pain. On the other hand, you can then image the animal again and again and again, potentially, if it's appropriate. So, it's high risk, high reward kind of stuff. Okay, so you've done your analysis. Sorry, you've done your imaging. You've collected these images, which are basically, it's a video, right, of the neurons getting brighter and dimmer and brighter and dimmer. You've probably collected it at about 30 frames per second. It could be 100 gigs that you've got. What do we do next? Well, our goal is we want to know how those neurons get brighter or dimmer over time. So we do sort of 3 primary steps. The first is called registration, right? So while this mouse, whether this mouse is anesthetized or not, the brain is going to move. It moves in response to heartbeats. It has a regular pulsation. If the mouse is awake, it's going to be jiggling all over the place anytime the mouse moves. Now, all over the place, right? We're talking 10, 20, 30 microns, but that is the width of a neuron. So you use an algorithm to basically register, to fix the frame so that from each frame to the next, all of the neurons are in the same place. Then you perform segmentation. So segmentation is the imaging equivalent of clustering, right? You want to know where the neurons are. So you can basically tell the computer, a neuron is here, so figure out how bright all of these pixels are of the image over time. You can do this manually, where you literally get the mouse and you draw a little shape around each neuron. Or there are automated segmentation approaches where you just get a computer and you say go. And there are pros and cons to each of those. And then finally, so you've got your movie, you've stilled it, you've put on the anti-shake, that's what the registration is. Then you've done the segmentation where you've said where all the neurons are. Now we've got to get the data out. And even that is still a problem. So this would be you'd be very happy if this is what you were going for. If this is what you're expecting, a beautiful two-photon image. Actually might even be confocal looking at it. Anyway, that's not the point. If this was your GCamp image that you saw down the two-photon, I would just say do this by hand, right? Just draw an outline around each neuron, okay? It's going to take you half an hour at the absolute most. You're going to do a perfect job. You're going to be absolutely confident. There are some pluses and minuses, even in a very small sample like this. Because the problem is, if you lose your data and somebody else comes along and does the same thing, right? You go away and the boss says, oh, we need that data back? And you say, oh, the hard drive got corrupted, just analyze the raw files again. They're going to come up with different numbers, right? Because their ROIs, their regions, their segments, the little circles they draw around the neurons are not going to be exactly identical. And that means different numbers. And that raises the possibility that somebody could analyze your same data and come up with a different result, which you do not want to happen, right? This is a big problem. You want your findings to be reliable and repeatable. And if you can't even repeat the result from the same data, we have a problem. Now, hopefully, as I say, in data this small with such perfect neurons, people are going to do almost the same outlines in every single neuron, but you never know. However, what about this, right? So this is cutting edge two-photon microscope. You can see each neuron going. What we have here is different focal planes through the cortex. So these are all different neurons. There are approximately 10,000 neurons in this. So my guesstimate is it would take you 4 working weeks, probably a little less. I'm being a little slow, but it could easily take you 3 weeks to do the registration for this. It's not the registration, sorry, the segmentation, drawing the ROIs, the regions of interest, drawing where each cell is. We don't have time for that. So we've got to get a computer to do it. And thankfully there are some good ones out there. Something called Sweet2P, which I've used, something called Cayman, a few others. And they're historically have been a pain to use. They're getting better, but the worst thing is they're still not perfect, right? So what you have to go through is you tell it to go, you come back an hour later, and then you have to go through and check them all, and that's still going to take you all day, just to check the computer. But still, a day is better than 3 or 4 weeks. Finally, we have signal extraction, right? Now, I've said that two-photon microscopes are great because you don't get out of focus fluorescence because of that fact that there's only right at the very focal point is there enough laser light to excite anything. It's unfortunately not strictly speaking true, right? You do get out of focus fluorescence and it can be significant. You get, a million times less than you would if you were using a confocal, but it's still there. And so this is what you can see here. So this blue signal is just the image from right on the center of a neuron. Okay, so it's right where the neuron is. The red signal is the signal taken around the image, but excluding all other neurons. And what you can see is that some of this blue signal appears to correlate with this red signal, right? And what's happening here is this is just when all of the neurons are active. So you need a way to subtract off that background signal, because otherwise you don't know if you see a calcium transient, whether it's your neuron being active or just the rest of the cells in the background being active. And then finally, after you've done all of that, you present the data as so-called delta F over F, which actually is the fluorescent signal at point T at at a certain time minus F naught, where F naught is some kind of baseline. Historically it was the fluorescent value at the beginning of the recording. That's where the zero comes from, the fluorescence at time zero. But typically now we actually do some minimum in a window surrounding the data. So the smallest fluorescence we've seen from this cell in the last 10 seconds, for instance. So it's the difference in brightness divided by the baseline. And that gives you the delta F over F. And then you also might typically apply some kind of smoothing, some kind of filtering to just smooth out the signal to make it look a little nicer. Unfortunately, this area is not very well considered. Sometimes people do it without really thinking about what they're getting up to. They just do it to make the data look nice rather than considering that it could come with consequences. But that's a whole nother story.